So hi, I'm Julia. I'm currently a fourth year and I'm going to give you your lecture on parasitology and mycology. So if you could all join this Socrative room, hopefully you all know about Socrative. If you just Google it, you'll find the website, and then this is the room number. So then we can ask questions as we go along to try and make it a bit more interactive. So I'll leave it there for a second. Okay, and then we're going to get started. So we'll start off with parasitology. So this is your syllabus, and it's actually quite short. There's not much on it, and you don't really need to know much detail in second year, so it's just having a vague idea of some of the key parasites that infect humans, I guess. So some definitions. So most of the parasites we'll be talking about are protozoa, which are unicellular eukaryotic organisms. And yeah, so that includes malaria. And then I'll also quickly discuss Helmands at the end, which are parasitic worms. So this table briefly covers the details of some of the parasites I'll be talking about today, including their host habitat, how they're transmitted, and the site where they tend to invade humans. As you can see, the skin is the most common invasion site for these parasites, as a lot of them are spread by an insect biting you, but Toxoplasma is spread by ingestion. And it's also important to notice that they do all infect different types of cells, and some are intracellular and some are extracellular, so just, yeah, I guess make sure to notice that. So this is the worldwide significance. So this is the WHO's top 10 causes of death in 2019. As you can see, the leading cause is ischemic heart disease, and then stroke is second, and there's actually only 3 communicable diseases in the top 10. But this changes quite significantly if you look at low-income countries, where infectious diseases become a lot more prevalent in the causes of death. So now the top 2 causes of death are infectious or communicable, and you can see malaria also making an appearance in the top causes. And this demonstrates why it's really important to understand these diseases and find better ways to treat and prevent them. This graph just shows death and DALYs, which are disability adjusted life years. So it just shows the impact that these diseases can have, even if they're not killing people. So you can see the top 5 causes, so the diarrheal diseases, TB, HIV, malaria, and hepatitis, actually are on a different scale, just because they have such a large impact. So yeah, again, this just really highlights the huge impact that these diseases can have on people's lives. Sorry. There we go. So first I'm going to talk about malaria, which is the only parasite that's on the left-hand side of your syllabus. So malaria is spread by the female Anopheles mosquito, and it is an apicomplexan parasite. And so it has an apicoplast, which is an organelle that's vital for the parasite survival. And the apicoplast hosts important metabolic pathways, such as fatty acid synthesis. And the apicoplast is quite an interesting organelle. It has 4 membranes, and it's often described as being similar to a chloroplast, which you get in a plant. And it's been proposed that Plasmodium got its apicoplast originally via endosymbiosis, that's engulfing another cell. And learning about this apicoplast is just important and interesting because it could be a really good target for anti-malarial drugs, especially as resistance to lots of the drugs we have currently is increasing. So there are 5 species of Plasmodium, which is the parasite that causes malaria, which are known to infect humans. The predominant species worldwide is Plasmodium falciparum, and that's the one people often mean when they refer to malaria. The different species all differ slightly in their life cycles. So, for example, Plasmodium vivax, which is probably the next most predominant after Plasmodium falciparum, has a dominant stage in hepatocytes in the liver, which Plasmodium falciparum doesn't have. But for the rest of the slides on malaria, I'll mainly be focusing on Plasmodium falciparum. This is a simple diagram of the Plasmodium parasite, showing you can see the cell entry machinery at the apical end with the polar rings. And then you can also see the apicoplast in there, and this figure on the left is from a paper looking at the 4 membranes of the apicoplast using cryo-electrotomography. So you can see they're highlighted. And this is the Plasmodium life cycle. So it's one of the most complex, I think, like definitely the most complex of the parasites you need to know. The life cycle involves both the host and the vector, and it consists of lots of different growth and proliferation stages. So in humans, there are 2 different stages, so the blood stage, and then the liver stage, and then the blood stage. And then in the mosquito, you get the sporogonic cycle. So it is quite useful just to learn this life cycle, I think, so you know all the different stages. But if you have to remember just some key stages, then it's important to know the form that's injected into humans by the mosquito, so that's the sporozoites. And then these infect the liver cells and mature into schizonts, and then which rupture, and it's then they release merozoites into the blood, and it's these merozoites that infect the human red blood cells. Then in the red blood cells, there's both an erythrocyte cycle and also a sexual erythrocytic stage, so which forms the gametocytes, and then it's the gametocytes that the mosquito ingests. And also it's the blood stage parasites that cause the clinical manifestations of malaria. So yeah, these are the important stages to remember. And this is just an image to show just how different the parasite looks in each of the stages. Yeah. So now for the first Socrative question. So I think this question should be there. Let's see if any of us answered it. No, 2 of you are logged on. Or if you just want to post it in the chat if you've not got on the Socrative page. Okay, yeah, of course. I think it was. I just posted the code in the chat for anyone who didn't get it originally. Okay, so yeah, you all got that right, which is good. You know the main form, and then another question. So, which is the form of the plasmodium parasite that is ingested by the mosquito? Okay. And yeah, so looking at the results, the majority of you went for gametocyte, which is correct. That is the form. So that's the sexual stages. So there's different ones. So there's a male and female gametocyte, and then these in the mosquito undergo sexual replication. So yeah, brilliant. So now looking at just the clinical manifestations of malaria. In uncomplicated malaria, which is when it's less severe, the classical malaria attack lasts 6 to 10 hours. So this consists of going from a cold stage to a hot stage, and then a sweating stage where people return to a normal temperature. And then these malaria attacks occur every second day with P. falciparum. Like a lot of symptoms you're told about, these actually are rarely observed, these attacks, and the sort of general symptoms of malaria is headache, vomiting, nausea, arthralgia, and chills. And that means that if you're in a country where you're not expecting to see malaria, sometimes these symptoms could be mistaken for flu. But in countries where malaria is prevalent, people often recognise them as the symptoms of malaria, so we'll be quick to treat it. Then severe malaria is a medical emergency. And this is when the infection of malaria is complicated by organ failure or abnormalities in the blood or metabolism of the patient. And there are lots of different manifestations of this, and I've listed some of them on this slide. So for example, cerebral malaria, which is when the parasitized red blood cells are sequestered in the brain in an interaction mediated by one of the parasite proteins, PFEM1 and ICAM1, which is on the vascular endothelium. So chloroquine is one of the antimalarial drugs that is used. And so this drug is a quinoline, and it accumulates in the acidic food vacuoles of the trophozoites, I hope that's the right way to say it, inside red blood cells. And then it prevents hemoglobin degradation from occurring. So the malaria parasite normally uses the heme as a source of amino acids, and it needs it for a food source. But heme is also toxic. So normally the malaria parasite would sequester heme in a biocrystallization process and form a large insoluble crystal called hemozoin. But the drug chloroquine stops this from happening. And this means that free heme accumulates inside the parasite. And the free heme lyses the parasite membranes and leads to the generation of reactive oxygen species, as well as causing other toxic processes, which would eventually kill the cell. So chloroquine is quite effective at killing the parasite, but unfortunately resistance to chloroquine is increasing, so now it often can't be used. So the resistance mechanism is that there's reduced accumulation of the chloroquine inside the acidic food vacuole, but I'm not sure we actually know exactly why that is, so whether it's reduced influx or if it's increased efflux via a pump or something. But yeah, so I've listed some other drugs that can be used to treat malaria on this slide. So you can see a lot of them target the blood stages of the parasite, and then primaquine is normally used to treat P. vivax rather than falciparum, as it targets the dominant form in the liver cells. And the drug used to treat malaria infection depends on the type of the species of plasmodium that's infected the person, the area where infection was acquired, and the drug resistance status of malaria in that area, and also the clinical status of the patient and pregnancy. So one of the ways malaria evades the immune system is by using antigenic variation. So when plasmodium invades the red blood cell, it modifies the surface of the cell, forming knobs on the red blood cells. So you can see those on the image in the corner. And these knobs on the surface contain parasite proteins, including PFEMP1, and they cause the red blood cell to adhere to vascular endothelium. So the PFEMP1, so the parasite protein, is encoded by VAR genes and serial expression of different antigenic types of these PFEMP1 enables chronic infection to be established, as if the antigen keeps changing, it's harder for the immune system to recognize and clear the infection. So this antigenic variation involves transcriptional switching among members of the VAR gene family. But yeah, different VAR genes will be expressed. But the VAR expression is stage-specific and monoallelic, which means that only one VAR gene will be expressed in each infected red blood cell, and a single gene is transcribed as the parasite matures through its life cycle. And this is important because the process needs to be carefully regulated, as the parasite needs to balance the need for safeguarding its antigenic repertoire, so it doesn't want to expose all of its antigens at once, because then the immune system would learn what they all are, but it also needs to change the antigens it expresses so that it can evade the immune system, if that makes sense. And so the antigenic variation, it's sort of regulated at several levels, including the DNA sequence elements, epigenetic factors, so the conformation of the chromatin, and then also the physical, like the nuclear position of the VAR genes. So a malaria vaccine, there's a lot of effort going into trying to develop one, but we're still not there, unfortunately. It's really complex because the parasite changes so much in each of the different stages, so choosing where to target it and how is quite complicated. But this diagram shows the stages where you could target a vaccine to try and prevent infection, and also some of the current trials that are ongoing for each of these different stages. And this review by Simon Draper is actually just quite good if you're interested in malaria vaccines. So now to move on to some different parasites. So the next one is Toxoplasma. So this is a bit different to the others because humans aren't actually part of the replicative cycle. So the main life cycle of the parasite actually occurs in birds, mice, and cats, but humans can become infected by eating undercooked animal meat or consuming contaminated food or water, and also transplacentally, so a fetus of a pregnant woman. In humans, the parasites form tissue cysts, which may remain throughout the life of the host. And generally, the infection is asymptomatic and doesn't cause a problem, but where it can cause issues is in pregnant women, as the parasite can damage the unborn child. And the damage is often more severe if the infection occurs earlier in pregnancy, and it can cause miscarriage, a stillborn child, or children may be born with signs of congenital toxoplasmosis, which can include an abnormal enlargement or smallness of the head as a child. So then next parasite is Leisioniasis. So this is a parasitic disease found in parts of the tropics, subtropics, and southern Europe. And it's caused by Leishmania parasites. And these parasites are spread by sandfly bites. The sandfly injects promastigotes into the human, which are then taken up by macrophages or other phagocytic cells. And then the promastigote transforms into a mastigote, which multiply in cells and infect other cells. So the sandfly actually ingests the infected macrophages, and then within the sandfly, the mastigotes transform into promastigotes, and that's the sort of complete life cycle. And both parasite and host factors determine whether the infection becomes symptomatic and what sort of infection occurs. So there are multiple forms of leishmaniasis. So you can have cutaneous form, which is where you get skin sores that occur like weeks to months after the initial bite, and the sores are usually painless. And then this can become diffuse, which is when you have the disseminated lesions all over the skin. And these are the treatments that are used, although these treatments aren't actually great, and yeah, they need to be replaced by other safer drugs. Then you can also get mucosal leishmaniasis, which sometimes follows on from the cutaneous form with certain forms of the parasite, certain species, sorry. And the parasite spreads from the skin to form sores in the mucous membranes of the nose, mouth, or throat. And then the more serious form of the disease is visceral leishmaniasis. This affects the internal organs, spleen, liver, and bone marrow, and this can be life-threatening and develops within months of the bite. And yeah, some of the symptoms listed there, so it's just not good. So the treatment for this includes supportive therapy, which means addressing the nutritional status of the patient, addressing anemia, and preventing secondary infections. And then also some of these drugs listed here. So moving on, sorry, we're going quite quickly through all of these. So then trypanosomiasis. This is caused by the trypanosomes, which are spread by the vector of the tsetse fly. And there are two main subspecies that cause disease in humans. Sorry, for African trypanosomes, there's TB gambiense, which causes a chronic disease, so West African sleeping sickness, and then another species that causes an acute disease. So the life cycle of this, so the tsetse fly injects metacyclic trypomastigotes into the skin tissue, which then transform into bloodstream trypomastigotes, and which replicate by binary fission. So the important thing to note about this parasite is, unlike the others, it's not intracellular at any point. So the whole life cycle is extracellular, which is different to all the ones we've seen so far. So now for another couple of Socrative questions. So sorry, I don't think I moved on. So yeah, which form of the leishmania parasite is injected into humans by the sandfly? This might be quite a hard question. The results. Oh, actually, yeah, okay. So yes, it's the promastigote that was just shown on the slide. So some of you went for sporozoite. That's not actually that's one of the malaria forms rather than a leishmania form. So that was a bit of a trick question, sorry. But yeah, so it's a promastigote. The malaria stages tend to end in this oit or oity, and then the leishmania is the astigote on the end. And then another question, what's the vector of the trypanosome? Okay. Okay. So yeah, the tsetse fly. I'm not sure I'm saying that right. Sorry. That's the vector of the trypanosome. The sandfly is for leishmania, and the mosquito is for malaria. So moving on. So yeah, one of the big problems with these neglected tropical diseases is that drug development happens very slowly, if at all. So we're actually still using very old drugs for these. There's not been as much development as there has in lots of other areas of medicine. So this image shows the drug discovery pipeline and highlights the 3 stages where drug development tends to stall. So the first gap is that the basic research is done in a lab, but then preclinical research isn't considered, so we never get to develop the drug candidates. The second stage is that the preclinical research is done, so we have the drug candidates, but then the big pharma companies or drug companies don't think there's going to be any profit from the drug, so there's no point in them to develop it clinically or take it forward. And then the third gap is that the drugs don't reach the patient. So even though the drug's been developed, it has too high a price, so people that need it can't afford it, or people don't produce it because, again, they don't think there's enough profit in it. So yeah, those are some of the key problems. Also, it can be quite difficult to develop drugs for parasites. As I said, with the vaccine for malaria, that shows that it can be difficult because the parasite has so many different stages and expresses different antigens and takes different form. And sometimes it's not in the bloodstream for that long. It may infect cells really quickly, so that can make it quite difficult. Some solutions to the problems are We could license veterinary drugs for human use, so we have drugs that can treat the disease in animals and they would be safe for humans. That can be a leap that can make it quite cheap to get the drugs for humans. Public-private sector partnerships to encourage investments in making drugs for these diseases. And then also drug donation programs can help, so when drugs are donated to the people that need them to treat the diseases. So there's a couple of examples for drugs for parasites that have been donated in schemes. So now to end the section on parasitology with a quick word on Helmands. So I'm going to talk about soil-transmitted Helmands, which are intestinal worms infecting humans that are transmitted through contaminated soil. So there's 3 major types that infect humans, and that's Ascaris, which is roundworm, and then whipworm and hookworm. The numbers in brackets next to them are the estimated amount of people worldwide thought to be infected. So as you can see, we never think about them in this country, but they are quite prevalent worldwide. So these helminths live in the intestine, and their eggs are passed in feces of infected individuals, and the eggs are then deposited in soil. And Ascaris and whipworm become infective as they mature in soil, and people become infected when eggs are ingested. So if they have contaminated dirt on their hands and put hands in their mouth, or by consuming fruit and vegetables that haven't been carefully washed. And hookworm is slightly different, so the eggs of hookworm actually hatch in the soil, releasing larvae that mature into a form that can penetrate the skin of humans. So the main form of transmission for hookworm is by walking barefoot on contaminated soil. And this map just shows where helminths are found worldwide. So, well, this shows children require preventative chemotherapy to stop them getting infected, but you can see that it's mainly areas with warm and moist climates where sanitation and hygiene are poor, where these infections occur, and they're considered neglected tropical diseases, so they inflict a lot of disability and suffering that can be controlled or eliminated quite easily. Heavy infections with helminths can cause lots of different problems, so including abdominal pain, diarrhea, blood loss, and in most severe cases it can cause physical and cognitive growth retardation, which is why it's important to stop them. And then just a quick word on Cryptosporidium, which is, I should maybe put it forward, this is a protozoan parasite, which is found in the small intestine. So you can see its little dots here. This is the epithelium of the small intestine, and the small dots are the parasite. So it forms highly resistant microbial cysts, and so can survive in water. So that's how people tend to get it. It's a waterborne disease. This disease most often causes disease in immunocompromised individuals, especially those with HIV. And yeah, the symptoms are watery diarrhea, nausea, and vomiting. So then I think that wraps up the parasitology section. We can move on to mycology. Again, there's not actually much on your syllabus for this. You just need to know about some of the medically important fungi. So this is the syllabus, which I'll try and cover. So first of all, fungi are part of the eukaryote domain. So the eukaryotes, and there's lots of different types. Yeast seems to be the main ones that affect humans and that you need to know about. And the defining features of fungi have the thick, rigid cell walls containing chitin, which is a polysaccharide. And they can be uni or multicellular or dimorphic. And dimorphic is when they can be uni or multicellular depending on the environmental conditions. So these are the medically important fungi in a table summarizing some of them. They're opportunistic pathogens, so they rarely cause disease in immunocompetent individuals, and it's when someone's already immunocompromised that they start being an issue. So as you can see, some of them are commensals, so Candida albicans is found in a lot of healthy people, and some of them are environmental, so Aspergillus and Cryptococcus. They're common in the environment, and they're all yeast or yeast-like, and now covers them in a bit more detail. So as I said, they mainly affect immunodeficient or immunocompromised individuals. You'll probably learn about immunodeficiency in other areas of your course, so I won't cover it here, but it was just a quick recap of some of the things that may predispose people to these infections and cause immunodeficiency. So Candida albicans is the most common fungal species that can cause human disease, and it can cause 2 main types of infection, so mucosal, so that's thrush or oral candidiasis, or systemic, which is when it's a more serious infection and it's invasive and disseminated. And invasive disease really tends to only occur in immunocompromised individuals, as the immune system can no longer control the C. albicans. Because if you remember, so it's a commensal, so it's found in the GI tract and the mouth and vagina. So the immune system can't control this, it can invade and cause disseminated disease. So it can grow as a yeast pseudohyphae and a filamentous hyphae, and which morphological form it takes depends on environmental factors and its genetic network. And the hyphae form can be a challenge for professional phagocytes. And hyphae can also express toxins. So this is just a paper of discovery of a fungal peptide toxin, candidialysin, which damages epithelial cells. And this may be what allows C. albicans to penetrate barrier tissues and establish infections. As this paper showed, strains lacking this toxin don't activate or damage endothelial cells, which is interesting because your syllabus doesn't really mention knowing about fungal virulence factors, but this is a good example of one. So invasive candidiasis symptoms vary depending on the site of infection, but can include blindness, lesions on the skin, fever, shock, and renal shutdown. So you can see it becomes very serious. And there's the treatments are amphotericin B and fluconazole. And there's it causes disease in immunocompromised individuals. So the typical sorts of patients you may see are in neonatal, so newborns, which is undergone surgery, ICU patients, so already unwell, long-term steroids, so that's immunosuppressive drugs, so organ transplant recipients likely to be on immunosuppressants, and then also post broad-spectrum antibiotics. So if you disturb the microbiome of your gut, where Candida albicans tends to be in normal individuals, if you then disturb that environment, it can cause this Candida to get out of control, so it can cause an infection. So now on to Cryptococcus. So this is an encapsulated yeast, and the capsule is anti-phagocytic. So it's a challenge for phagocytes. Cryptococcus is a facultative intracellular pathogen, so it can remain latent in phagocytes, and that's how it spreads around the body because it can't be killed by them. An important thing to note about Cryptococcus is that it can be stained with India ink, so the ink can't enter the capsule that surrounds the cells, so you get a clear halo forming around the cells, which is shown in this image here. And ignore that they're pointing at the yeast budding, so that's the replication, but I just think this image is good to show the clear halo sign. So the people likely to get an infection with Cryptococcus are organ transplant recipients, anyone on long-term steroids, and HIV, especially if their T cell count has dropped really low. The symptoms tend to be a pneumonia, a cryptococcoma, which is a large fungal mass, so an infectious granuloma that typically happens in the brain, but can also be found elsewhere, so in the lungs, for example. And the big problem with the risk with Cryptococcus is that it can enter the central nervous system, and then it can cause meningoencephalitis, which is extremely serious. So it's important to treat the disease before it progresses to this stage. So yes, I think now for another Socrative question. So which of these isn't true of Cryptococcus? So yeah, so I actually think maybe I didn't explain this that clearly. So a lot of you got up for the right. So the right answer was A. It is not commonly found in the human intestine. That would be Candida albicans. So Candida albicans is a commensal, so it's found in the intestine and the mouth and vagina, whereas Cryptococcus is a environmental fungus. So people tend to get infected by breathing it in. So yeah. The next, Aspergillus, is another medically important fungus. So again, this is an environmental one. So Aspergillus species are ubiquitous in the environment, they're everywhere, and it's a filamentous fungi. And most people don't get infected because even though we breathe it in, our immune system is able to prevent infection, but immunocompromised people are more likely to be affected. And Aspergillus causes aspergillosis, and invasive Aspergillus has quite a high mortality rate, so over 25%. So people that tend to be affected are those with damage to the lungs, so like COPD, TB, or bronchiectasis. Neutropenia can cause an issue with Aspergillus, and then again, just other causes of immunosuppression. So aspergillosis, the disease human gets from an Aspergillus infection varies widely depending on the immune status of the person. So it can cause lots of different forms of disease. So non-invasive aspergillomas tend to occur in someone that has pre-existing lung cavity, so someone maybe that had TB already, and then is repeatedly exposed to aspergillosis may get one of these aspergillomas. And you can see there's a characteristic sign in the lungs of someone with aspergillosis, which is this crescent sign. So you can see in this image around the mouth there's a thin crescent. And then a worse form, so invasive aspergillosis occurs in immunosuppressed individuals, which were the conditions I listed before. And neutropenia can cause a big issue, as in neutropenia, people get excessive hyphal growth and the fungi disseminates throughout the body, so it can make people really ill. And then in other people, may have a hypersensitivity to the fungal components of Aspergillus, so they get allergic bronchopulmonary aspergillosis, and this can cause altered lung function. So it's just an example of how disease people get is really influenced by host factors as well as the fungus that's infecting them. And so next fungus we're going to talk about is Pneumocystis jirovecii. I think that's how you say it. So this causes pneumonia only in immunocompromised individuals. It's normally found in the lungs of lots of healthy people, but only tends to start causing an issue when people are immunocompromised. So the symptoms of a pneumonia with this fungus are a non-productive cough, weight loss, night sweats, and you can see the widespread infiltrates in the chest x-rays in the lungs. So it's diagnosed by a bronchoalveolar lavage, and then it's got characteristic cysts, so you can see a cyst on this chest x-ray here. And also when it's stained with silver stain, it has a crushed ping pong ball look, so the cells look like helmets or crushed ping pong balls. So if you can persuade yourself, you can see that in the image at the top. And it's treated with high-dose cotrimoxazole. So I just thought I'd outline, I mentioned that they happen in immunocompromised people because they're opportunistic infections, but there's a big problem with fungi in HIV patients. So Candida albicans tends to be one of the first opportunistic infections that happens in people with HIV/AIDS. So it's one to look out for. And then, although mucocutaneous candidiasis is common in people with AIDS, invasive candidiasis in the more serious form is actually unusual unless other risk factors are also present. And pneumocystis pneumonia has classically defined AIDS as a syndrome, as it tends to occur when an HIV positive individual has progressed to AIDS, so can no longer control this fungus that might be present in their lungs. So that's actually all I had, so this lecture's been a bit shorter. If anyone has any questions, I'm happy to try and answer them, or feel free to contact me. My email's on the slide.